What are the new technologies that the HPRC is developing, and how are they transforming genomics? So I think the main thing that we're doing is proving that you can automatically assemble genomes at scale. And automated assembly is a bit of an overstatement maybe, because there's still a lot of human intervention, quality control, these kinds of things happening. But we basically are to a place with the project where we are doing incredible data production. So the initial sequencing data is of the highest quality using the newest sequencing technologies that are available that frankly are kind of a demonstration of a total revolution in nanotechnology, where we're able to observe single DNA molecules, many single DNA molecules in parallel and at very long lengths, very high accuracy, very low error rate. And because of this, and because of innovations in sequence assembly, we're able to then derive complete assemblies of many human genomes for the first time at a truly complete level. So these assemblies span effectively from one end of each chromosome to another. And they're not perfect in this particular iteration, but they're approaching a level of perfection, of completeness, that's never been seen for hundreds of, eventually thousands of assemblies. So in result, we have this incredible foundation on which to do genomics that didn't exist before. And in response to this, because we could see it coming for a while, that these technologies would enable this, a lot of members of the HPRC have been developing information technologies that work downstream of these. They make it possible to relate these sequences to each other. We call that alignment. To build representations of the many-way alignment between these genomes. We tend to call these graphs, pangenome graphs. And then we reuse these to do analyses, to understand evolution, of parts of the genome that weren't known before. So I guess that gets to the second part of the question, is how is this transforming genomics? And at the moment, it's subtle. So if you go to big genomics conferences, you do see use of these resources, but it's sort of the tip of the iceberg, I think. We're just beginning to see uptake in the biomedical community, in human medicine, and also in biology in general. In some species, people really need a pangenome in order to operate because there's high diversity, because the genomes they're working with are very different than each other. In humans, we've been able to get by on this kind of crutch that everything is kind of like the reference, and we just have to have one reference that represents the whole genome, and we can align short reads against it and understand lots about biology. But what we've shown in this project is that that is not the case. There are parts of the genome that were invisible. We've made them visible, and using that, we've solved basic questions in chromosome biology, for example. Recent work that I've been involved with demonstrated the reason that 1 in 800 people is born with a translocation between two chromosomes that connects them into a fusion. It's called a Robertsonian chromosome. This can impact fertility, and it's really big. So we've seen it for 50 years or more on cytogenetic microscopes. And only now we understand why it's happening, because there are sequences that are homologous, they're similar to each other on these chromosomes that recombine in this way, that foster this recombination. We saw that in the Human Pangenome data for the first time. And because of that, we're able to actually confirm that this was the case. So this is one small but textbook altering example of the way that our work is having an impact in genomics.